All right, now let's look at the hormonal control of the GI tract. I created a table here to show the several hormones. My concentrations are majorly on four hormones: gastrin, GIP, secretin, and CCK. We have more, there's multiline and the others, but I want to talk about this four in this lecture. And to understand this, I kind of created a flow of how the release happens and how they actually function. So we'll start from the stimulus, the cell stimulated, the hormone released from those cells, and the target cells and the functions. Let me create a blank sheet and explain what I mean. So on this blank sheet, I want to just explain. So this is stomach. I'm very bad at drawing, so you guys will bear with me. The stomach. It's not mine anyway, Sha. Okay, I just want to draw something simple to explain, just like this, and the intestine, and so on and so forth. All right, let me make this the bolus of food that we eat. So, the very first contact of food is after it passes the lower esophageal sphincter. At this point, I'll just call it hypothetically. The bolus of food comes into the stomach, and there's something it causes. It causes distension of the stomach. So as food enters here, the stomach kind of expands. So the distension of the stomach stimulates some kind of cells. The cells are present in the antral portion of the stomach, the duodenum, and the jejunum. The cells are called G cells. So G cells, let me write that well, sorry. So G cells get stimulated. Let me write the G cell properly. So G cells get stimulated to release gastrin. So gastrin, upon being released, its aim is to deal with this bolus of food that has entered into the stomach. So gastrin goes to some other cells present in the stomach called parietal cells to cause the release of acid, and also stimulates enterochromaffin-like cells to release histamine, which in turn goes to the parietal cell to cause the release of acid. The whole essence of gastrin release is to deal with this bolus of food that has entered into the stomach. I'll explain in details later. So after much activity of acid and also pepsin action on some proteinous compounds present in the food that entered into the stomach, the chyme is then released. Let me draw the pyloric sphincter here. The chyme is then released. So let me go back to the green color. So chyme has been created here, sort of. Chyme is now released into the duodenum. Now, when chyme comes into the duodenum, there are some certain cells that respond to chyme. So chyme has majorly two parts. So this is chyme. Chyme has the acid part of chyme, and then we now have the food material part of chyme. So the acid part of chyme, the cells that respond to the acid part of chyme are called S cells. S cells. While the food material part of chyme, two cells can respond to it. We have K cells, and then we also have I cells. S cells bring out secretin. K cells bring out GIP. While I cells, they bring out CCK-PZ. So when the acidic content in chyme gets to the duodenum, cells present in the small intestine, the duodenum, the jejunum, and the ileum, there are S cells present there. They release secretin, whose function we'll discuss shortly, and also K cells respond to the food part of chyme to bring out GIP, and I cells respond to the food part of chyme to bring out CCK-PZ, and CCK-PZ stands for cholecystokinin pancreozymin. So let's go to the previous slide to explain the functions now. Now, back to this. The stimulus, as we said, starting from the stomach, Distension of the stomach by food or peptides in the stomach stimulates G cells, which are present in the antrum, the duodenum, and the jejunum, and G cells bring out gastrin. Gastrin then goes to enterochromaffin-like cells, that's ECL cells, to stimulate, this plus sign stands for stimulate, to stimulate the release of histamine. And they also go to parietal cell to stimulate HCl release from the parietal cell, that's the proton pump. The mechanism of actions we'll discuss in details in acid peptic diseases. Now, fats, carbohydrates, and amino acid in chyme, once it's emptied into the small intestine, K cells present in the duodenum and the jejunum respond to it and brings out GIP. GIP then goes to G cell to inhibit the release of gastrin to reduce the amount of HCl that is meant to be released from parietal cell. It's understandable. We've just emptied food content into the small intestine, and we don't want much to come back into the small intestine to prevent acidic corrosion, if I could call it that. Now, the third stimulus is chyme, the acid part of chyme entering into the duodenum. Some cells tend to respond to the acidity of chyme, and those are called S cells. S cells are present in the duodenum, the jejunum, and the ileum. They bring out secretin, and secretin goes to the pancreas and tells the pancreas it's as if acid is in the duodenum. Will bring out a buffer, that's bicarbonate. Let us get this buffer to buffer the acid inside the small intestine, since the small intestine wasn't made to accommodate such acidic content. So the pancreas releases bicarbonate to neutralize the acid present in chyme. It also goes back to the stomach and kind of causes the acid in the stomach to be used up. Don't forget, pepsinogen, it's converted to pepsin by the help of this acid. So there's increased pepsin formation, which means the acid present in the stomach is used up, caused by the action of secretin also. And the last one we'll talk about it's cholecystokinin pancreasamine, but let's check the stimulus that brings it out again. So the chymes, fat, and amino acid in the duodenum stimulate I cells, which are also found in the duodenum, the jejunum, and the ileum, to bring out cholecystokinin pancreasamine, sometimes just simply called CCKPZ. So from the name cholecystokinin pancreasamine, cholecys is the gallbladder, so it goes to the gallbladder, and the pancreasamine, it goes to the pancreas to cause release of pancreatic enzymes. So let's look at that at the target cells and the functions. So the exocrine pancreas is a target for CCK, and it causes increased enzyme secretion from the pancreas. It also goes to the gallbladder and causes increased contraction of the gallbladder, then also goes to the sphincter of Oddi. Let me just explain that briefly on the next slide. Let me create a new one. So if this is the gallbladder opening like this is the second part of the duodenum. The gallbladder opens into here, then we have the head of the pancreas somewhat here. I'm bad in drawing, just bear with me. So the pancreatic duct kind of also opens here, bringing the pancreatic enzyme. Now there is a duct, there is an opening here, kind of called the sphincter of Oddi. So let me just call it here like this, the sphincter of Oddi. So CCK PZ goes to the gallbladder, stimulates the gallbladder to contract, to release biliary content out. Then it goes also to the pancreas to cause pancreatic enzyme to come into the duct to come out here. So they meet at this junction, the sphincter of Oddi. So CCK also causes the opening of this sphincter so that content of the pancreas will come into the small intestine, and content of the gallbladder, that's the bile, will also come into the small intestine. So back to our slide here. So that's what it does. And also at the level of the stomach, CCK causes decreased gastric emptying to prevent excess content of the gastric digesting product coming to the small intestine. So this simply explains GI hormonal control. Thank you.